Hello and welcome to Biocapsules. Here in this video, we'll be talking about another method of gene transfer in bacteria, that is transduction. So let's start with the topic. So, transduction is a method of a gene transfer where one bacteria transfers its genetic material to another bacteria with the help of a virus, right? Now we might have heard about many type of viruses such as plant viruses, animal viruses, but there are several viruses which particularly infects bacteria, and such viruses are known as bacteriophage. In short, phage. So the transduction is a process where the transfer between the bacterial cells occurs with the help of a bacteriophage. Now to understand this transduction process, let's first discuss the life cycle of a bacteriophage. Okay. So whenever a bacteriophage infects the bacterial cell, it may either show a lytic cycle or a lysogenic cycle. So first start with the lytic cycle. In a lytic cycle, when a bacteriophage infects the bacterial cell, it injects its DNA, that is viral DNA, into the host cell. Now this viral DNA synthesizes some protein which degrades or breaks down the bacterial genome. Okay. Now this degradation of the bacterial genome allows the bacteriophage to take the control over the cellular machinery of the bacterial cell. Okay. Now when the bacteriophage takes the control over the cellular machinery of the bacterial cell, it starts producing new phage particle in the bacterial cell. Okay. So during this process, the host cell become gradually weakened by the phage enzyme and eventually burst out, releasing new phage particle in the surrounding environment. In this way, the infected bacterial cell produces new phage particle, and these new phage particle then reinfects other bacterial cells in the environment. Now let's see what happens in lysogenic cycle. So in lysogenic cycle, when a phage injects its DNA into the host cell, this linear phase DNA becomes circular and gets itself integrated into the bacterial genome by recombination, where this viral DNA stays silent or dormant for many years. Now this integrated phase DNA is called prophase. Okay. Now, every time when this host cell divides, this viral DNA also gets replicated along with bacterial genome and transferred to the progeny cells. Now, if by any chance this bacterial cell gets exposed to some mutagenic agents such as UV radiation and certain chemicals, this viral DNA gets activated and it excises itself from the bacterial genome and becomes circular and starts lytic cycle from this stage. Okay. So the major difference between the lytic cycle and the lysogenic cycle is, in lytic cycle, the host cell is died at the end of a process, whereas in lysogenic cycle, the host cell remains alive. Now, based on which life cycle the bacteriophage chooses, there are two types of transduction. One is specialized transduction and another is generalized transduction. So first start with the specialized transduction. In a specialized transduction, the bacteriophage undergoes a lysogenic cycle. So as in a lysogenic cycle, when a bacteriophage infects the bacterial cell, it injects its DNA, that is viral DNA, into the host cell. Now this viral DNA gets itself integrated into the bacterial chromosome by recombination and form a prophase. Now if this viral DNA gets activated, it breaks off itself from the bacterial chromosome, and during this process, few genes of the bacteria also remains attached to it. Now these genes keeps on replicating along with the viral DNA and becomes a part of a phage particle. Now when such phage particle infects any other bacterial cell, then these genes present in the phage particle are also transferred to the new bacterial cell. So the important points about specialized transduction is it involves the lysogenic cycle, and in specialized transduction, the bacteriophage contains both viral and bacterial genes, and this transduction is called specialized because only some specific genes of the bacteria are transferred to the another bacteria. Okay. Now next is generalized transduction. In a generalized transduction, the bacteriophage undergoes the lytic cycle. So as in a lytic cycle, when a bacteriophage infects the bacterial cell, it injects its DNA, that is viral DNA, into the host cell. Now this viral DNA synthesizes some protein which degrades the bacterial genome. Okay. Now this degradation allows the phage to take the control over the cellular machinery of the bacteria. It starts synthesizing new phage particle. Now during phage assembly process, the phage mistakenly packages the bacterial DNA into the viral head instead of a viral DNA. So this results in the formation of a virus particle containing bacterial DNA. Now when this phage infects a new bacterial cell, it injects bacterial DNA into the host. In this way, the bacteriophage transfers the bacterial DNA from one cell to another cell. Okay. So the important points about generalized transduction is it involves the lytic cycle, and the bacteriophage in the generalized transduction carries only the bacterial genes. That's all in this video. If you like the video, hit the like button, share with your friends, and do subscribe my channel.